Well, good afternoon, everyone. Welcome to Spain for those that you came from the foreign. Well, first of all, I would like to thank to the organizers for the invitation that they gave to me, and I always enjoy a lot to talk in this kind of forum and discussion groups. So before anything, I would like to say that I seldom use PowerPoint slides to do my talks, so forgive me if my talk goes differently than the PowerPoint slides, okay? So the reason I do that always is that I'm very glad if you can make me questions during the presentation, so don't be shy, sorry, and let's go, okay? So well, as the chairman was saying, my name is Ignasi Belda. I'm the CEO of Intelligent Pharma. Intelligent Pharma is a company that I founded myself like almost 9 years ago here in Barcelona, and what we do is computational chemistry. We are a company that we provide global services for pharmaceutical companies worldwide, and our expertise is in computational chemistry. We have a big group of computational chemists, bioinformaticians, biochemisticians, etc., and they are mainly using tools that we self-developed to be able to predict some things and some points for our clients that we'll explain in a minute. So, well, first of all, we'll take advantage of this talk to do a really brief introduction to the company, so you will understand the context where we are working. We have several business lines, but the business line where I'm going to focus today the talk is in the first one, in our molecular modeling services. So as I told before, we are a service company, and our services are services based mainly on artificial intelligence and supercomputation. So nowadays we have clients, as I said before, worldwide. We have clients in the US, in Asia, obviously plenty of places in Europe, etc. And we have 5, here there is one missing, 5 offices worldwide. Our headquarters are here in Barcelona, just 100 meters away from here, but we have also offices in Germany, UK, US, and Canada. This is the kind of projects that we usually do, right? I guess that you are not experts in the pharmaceutical field, so probably this doesn't mean anything to you, but probably at the end of my talk you will understand some words that you are seeing here. So, for those of you that you are not familiar with the pharmaceutical industry, what I would like to say is that the pharmaceutical research is really a long process. From the beginning to the end, normally there are 12 years, right? So here at the really beginning is where we start on researching a new drug, and at the end is where that drug comes into the market. So as I told, the time to market is around 12 years on average. The cost is around 1,000 million of euros, or 1 billion American billions, okay? And another important thing is that only one project amongst 15 that pass through this point in here, okay? From this point, only one amongst 15 is able to reach until the end. So this is devastating, because you can imagine that the investment that the pharmaceutical companies are applying to develop a new drug in here is huge. So only one amongst 15 comes into to the end. So as you can guess here, this is the most hard step, and this is the clinical trials where you can test the drug in human beings. Before that, the human beings are not used as a test. Yeah.
The terms hit to lead, or what's the layman's explanation of those?
Well, there are a lot of explanations. One of them is toxicology, because when you are developing a new drug, you are making toxicology predictions with in vitro models or animal models, but obviously the human biology is very different from a monkey or a pig or a dog or a cell, right? So a big number of projects fail because of toxicity. So that's one reason. Another reason, and this second reason for me is very sad, is mergers and acquisitions, because these huge pharmaceutical industries, they are researching, I don't know, in cancer, and suddenly they merge with another giant, right? And the second giant is also researching on cancer. So then they have to cancel any of the two projects because they are not going to compete internally. So that's a sad reason because they are canceling projects that they are competing amongst each other. There are more reasons, right? But toxicology and strategical reasons are two of the main reasons.
Basically, the word HIT is the acronym or what's the
word HIT?
Yes.
Sorry, I misunderstood your question.
Yes.
HIT here. In 2 slides, I will explain what a HIT is, okay? So please wait just a couple of minutes and I will explain it. Anything else? It's great that you can ask questions because then it will be more than a dialogue, more than a monologue. So, as I thought at the beginning, we have a relatively big number of engineers and mathematicians developing new tools, and those tools are the tools that we are using internally to provide services to our clients, right? So the major part of those tools are using artificial intelligence, but those tools here are using, in addition, high HPC, right? Supercomputing power. I'm not going to explain all of those tools because that would take long, and I think it's not the moment now. So I will focus the presentation today on this tool, which is called Mobius. Mobius is the fruit of a collaboration between us and an American company, and well, I will explain in a minute. But before that, let me explain what a HIT is, right? In this slide here, I'm really oversimplifying the problem, right? And what I'm going to explain now in a couple of minutes is all this from here to this point, okay? Not the human trials, right? This step of here, all those steps in there, right? So that can take like 6 years or something like this, right? So the first point, the first seed is where the pharmaceutical company says, okay, I want to target this protein. So the target normally is a protein, and what we want to achieve is to design a new compound, a new chemical compound that is able to target this protein because this protein is doing something bad. Like, I don't know, for instance, is helping to the cancer cells to go out of the tumor and infect other tissues, right? So if we discover how this protein is acting, we can then design a new drug, a new chemical compound that goes there and tries to block the action of this protein, right? And that's the hit identification. The hit identification is, right, we are going to screen hundreds and sometimes millions of random compounds to see if we are able to find a hit, right? A chemical compound that presents some desired therapeutic activity, right? So the formal definition of hit would be a chemical compound that is able to do what we want to achieve at the end. Probably this hit is a bad drug because probably it is toxic or is not soluble or is very expensive or whatever. But a hit is the first compound that we are going to use as a seed for the future drug, right? So next step is to find the lead. So from the hit to the lead, we have this step called hit to lead. And here we have a process of optimization, right? So it's an optimization where some guys called medicinal chemists, they like to change some chemical groups of this compound, of the hit, to allow the heat be less toxic or more cheap or more soluble or whatever, right? So it's a bit, for me, it's unbelievable, but nowadays in the major number of pharmaceutical companies, this step is done by hand. I mean, in a manual way. Almost, well, a very few number of companies are doing this step by using computers and simulations and these kind of things. They do it just by hand. For me, it's like, I don't know, designing the aerodynamics of a new airplane by using manual stuff and using your own calculator instead of using simulators of the aerodynamics, right? So that's what we do. We have designed a lot of tools, computational tools that use artificial intelligence and supercomputation to achieve this part of the heat identification. And also, we have developed other tools to work in the heat to lead part. Okay? So once that we have the lead, the next step is the lead optimization to get the drug candidate. And then when we get the drug candidate, then we can go to human beings, to see if that drug candidate is really performing better than the placebo or not. So the tool that I wanted to explain to you today is a tool that is able to work in this step, but also in this step, in both, right? Is, as I told you before, Mobius. So I don't like too much this slide, there is too much text, please avoid reading that. Mobius, in very few words, is an evolutionary algorithm. Probably most of you are familiar with genetic algorithms and evolutionary computation, right? So I don't have to explain too much about that. But in few words, genetic algorithms are a kind of algorithms that generate a population of random individuals, in this case compounds, right? Then we evaluate individual by individual, compound by compound, which is their fitness, and then based on that fitness, then we generate a second generation of compounds, and so on and so forth. So then we evaluate the second generation, we evaluate them, then we get a third generation, and so on. So as you can imagine, the problem is really complex, because the number of compounds that could act as a drug is higher than the number of atoms in the universe. So it's not an easy task to be able to find a hit, because the number of combinations is, as you can imagine, well, almost infinite. So to achieve this step with certain success, we need supercomputers. Otherwise, it would be impossible to perform this step with a single machine with few cores and nothing more. We have to test really huge populations of individuals, test them, and then iterate until we find any optimum. And of course, we are aware always that with genetic algorithms, it's hard to find the global optimum. But we sometimes are happy getting a local optimum, because then we optimize that local optimum and perhaps we get a good drug, right? So, as you can imagine, the problem of this context is not the genetic algorithm per se, is the evaluators. The problem is, are all those softwares or all those programs that we are using in combination with Mobius to be able to evaluate the fitness of the candidates that Mobius is proposing, right? So today I'm going to explain just three of them. I don't want to enter into technical details, and probably this is the less technical talk that you will listen today. Forgive me about this, but if you have questions, technical questions, we can chat about that. But those three tools or softwares or technologies, whatever you want to call it, that collaborate with Mobius to evaluate the fitness of every compound are Selene, Hercules, and Pegasus. And depending on the information, the prior information that we have about every project, we are using one tool or the other, or a combination of the three. So the first tool is this one, Selene. Selene is what we call a receptor-based virtual screening tool. What does it mean? That means that if we have the chemical structure, the 3D structure of the target protein, okay, with Selene we can make the simulation of how the drug candidate or the heat, the future heat or the heat candidate, whatever you want to call it, is able to dock into the protein, and once that we discover which is the structure of this docking, okay, we can get the energy, the interaction energy between the candidate and the protein, and by doing this we can discover if that can be a good candidate, a good individual or not, right? So in one of the previous talks, it was mentioned GROMACS and NAND and this kind of tools. Those tools are about molecular dynamics, right? Probably you are familiar, some of you are familiar with molecular dynamics. The big difference between molecular dynamics and docking is that molecular dynamics is like making a film, and this is like making a picture, like a photography, right? So as you can imagine, molecular dynamics is much more expensive from the point of view of computer power than this. But nevertheless, this is also very expensive because it takes like, I don't know, a regular experiment takes 30 minutes. A regular docking experiment with Selene takes 30 minutes. So if we are analyzing a population of, I don't know, 100,000 compounds, then it's 100,000 times 30 minutes. So this is the explanation why obviously we need in a mandatory way supercomputers. So by using Selene, once more, what we do is to try to simulate how the compound is going into the protein, how is matching that protein, and by doing this we analyze the energy, and then we discover which is the fitness of that individual to be able to help to the genetic algorithm to evolve and propose the next generation. Next tool is Hercules. Hercules is a tool that normally when I explain that tool to academics, they're really surprised because one thing that usually happens in academia but doesn't happen in industry is that when we are designing a new drug, sometimes in the industry, sometimes it's 50% of the cases, we don't use a protein. So the target protein that I was talking at the really beginning, this one, it doesn't exist. So normally when we are designing a new drug in the industry, we really start from here without having any idea of which is the target that we are trying to approach, right? So to be able to be successful with this kind of projects, we use Hercules. And Hercules is a ligand-based virtual screen tool. That means that what we are trying to do is to analyze if a new drug will be a good drug or not, a good hit will be a new drug or not, depending on their molecular fields. Okay? That's a conception a bit abstract, but okay, imagine that this is a new drug, right? A new compound. So here, I'm going to use this microphone because I need two hands. Can you listen to me here? Yeah. So imagine that you have this is a new drug, okay, and here we have a positive charge. So here there is like a positive field, okay, that is surrounding this positive charge. So imagine that now here we have a hydrophobic area, an area that is rejecting water. So here we have a kind of area that is rejecting this water. So this is called molecular fields. So what we do is, okay, we take one compound that is working, that is doing the therapeutical effect that we want to achieve. We calculate those fields, those molecular fields. We remove that chemical structure, and then we try to see if another completely different compound, like could be this, is able to match inside those fields, right? And that's what we are doing in Hercules. See, we have one compound, and these are the molecular fields of that compound. We have another completely different compound with other molecular fields, and Hercules is trying to look for the best superimposition. So the hypothesis behind this is that if two completely different drugs or compounds present similar molecular fields, then the therapeutical action or the biological action would be pretty the same. Okay? So imagine here we have two similar compounds, the green one and the blue one are quite similar, but the molecular fields are really different. So Hercules will not be able to match these molecular fields. So we know that this drug or this compound will not act like the blue one, right? So again, this is a really hard problem because we not only have to find the best conformation in the space, but also compounds are flexible entities. So if there is a bond that is spinning around, then all the molecular field is turning around. So it's a really complex problem. So again, when we do this experiment, this simulation per compound, it takes a long time, and then we have to multiply this time the number of individuals that we have in the population of the genetic algorithm. And finally, the last tool is Pegasus. Pegasus is a more cheap tool, and is the tool that we use when we have to screen really big number of millions of compounds. And this is not based on 3 dimensions, but is based on fragments. That means if we know that this drug is, I don't know, doing whatever we like to do, then we fragment the compound into several compounds, and then we analyze a huge library of millions of compounds that could mimic the behavior of our drug, and then we try to see if they share fragments, if they share chemical fragments. So by doing this, we can predict with quite a relatively high accuracy if 2 molecular compounds can be doing the same biological action. So that's the third tool that we combine with Mobius when we have to screen really huge libraries of experiments. Okay, so summing up, the tool that I wanted to present to you today was Mobius in combination with these 3 tools that are 3 tools that in a mandatory way they need supercomputers to run, because although we can take only 30 minutes to do an experiment here, but the point is that Mobius needs thousands of experiments of 30 minutes each. So then we need a big power of computers. What else? And that's all. Here we have some public clients. We are working for a big number of pharmaceutical industries worldwide, so unfortunately they do not allow us to explain that we are working for them, but some of them are kind, and they allow us to tell at least that we are working for them. And here you have my contact information with my email and the addresses of our offices. If you have questions, I'm very glad to answer them. Yeah.
Is it pretty typical for what you're doing for pharma companies to use like third-party specialists like you, or
can you repeat? Sorry.
Is it typical for pharma companies to use a service like yours, or is a lot of the work that you're doing done internally at like the bigger pharma companies? Or are you bigger customers and everything?
Some of our clients, they have internal departments that are doing exactly the same than us. Okay, so nevertheless, some pharmaceutical companies like redundancy, so they have internal departments, but also external companies like ours doing the same thing. But there are also that, unfortunately, due to externalities in the field, they are cutting R&D internal research, and they are outsourcing a lot of the research. And thanks to this situation, our company is growing because we are getting big clients thanks to that. Sure.
Do you have your own systems?
Yeah. At the very beginning, when I founded the company, we were users of the services of the BCC, especially Mare Nostrum. But nowadays, we have our own facilities, but also nowadays we outsource a lot of calculations, because when we have peaks of work, unfortunately our internal facilities are not enough, so we have to purchase external power to be able to deliver in time for our clients.
How big are your internal systems?
I don't know. Sorry. I can check with my colleagues later by WhatsApp. But I honestly,
obviously not as big as like BCC.
No, I don't think so. Probably it's 1/10 of BCC. But I will check later if you wish. But nowadays we are using a lot Amazon. So Amazon is a good tool for us, especially for those days where you have a huge peak of things to deliver and few days, so we go there.
Okay.
Anything else? Right, thank you.